And past natural selection has been constrained by various trade-offs. Now, some of those trade-offs also apply to us. The trade-offs associated or that result from conservation of matter, for example, mean that if plants are nitrogen limited and we select for higher protein seeds, we're also selecting for lower yield. And that's the same constraint that would have been faced by natural selection. But there are other cases where either our goals are different or, in some sense, the trade-offs that constrained past natural selection don't need to constrain us. And these are the cases where I've said that these past trade-offs present us with opportunities for present improvements. And I'll give 3 examples of that. So I'll just start with the power of natural selection in an agricultural example. Watergrass is a serious weed of rice, and we're all familiar with the ability of agricultural pests to evolve resistance to toxic chemicals we use to control them. Here we have watergrass from 2 farms, obviously resistant to this herbicide, and over here resistant to a different herbicide, whereas in the front we have watergrass from organic farms where they hadn't been exposed to herbicides, and that watergrass is still susceptible. So this is, I think, very well known, but it's important to remember that the ability of pests and weeds and so on to evolve resistance to our control methods apply to all of our control methods and not just chemical control. So watergrass in particular is known to be descended from barnyard grass, evolved over the last few thousand years in Asian rice fields. And farmers with hoes imposed strong selection against plants with red stems, which were easy to spot in the field. And as a result, watergrass evolved green stems that make it harder to distinguish from rice. And Spencer Barrett took this physical similarity or visual similarity further, looking at a bunch of traits that could be assessed by a farmer with a hoe, and found, in fact, that watergrass resembles rice more than it resembles its own ancestor, barnyard grass. And that's, in a couple thousand of years. Now, barnyard grass and many other weeds are readily killed by flooding. This is a trial run by UC Davis, and these are the deep water plots here with not much in the way of weeds. But watergrass has evolved parenchyma, these air-filled channels in its stem, which transport oxygen down to the roots, so it does just fine in flooded soils. So it's evolved resistance to farmers with hoes, chemical control, and flooding. And whatever else we use on it, it's probably going to evolve resistance to those as well. Something to think about. So that's sort of, the power of natural selection. But there are limitations. And my central hypothesis and one of the themes of my forthcoming book and also in our 2003 paper is that natural selection is unlikely to have missed improvements that meet all 3 of these criteria. That is, that they would consistently increase individual fitness. We're, of course, talking about past environments. That would require only minor genetic changes. So I'm talking about things like increased expression of an existing gene or changing a few amino acids at the active site of a gene, as opposed to something we were talking about in our agricultural discussion group, the possibility of designing enzymes de novo from scratch. There you might come up with something that natural selection had never seen before, and so it could be great, perhaps. But I don't expect that to happen very soon. And finally, that are trade-off free. So an example of a trade-off that I wouldn't have necessarily predicted from first principles. We know that the rubisco, the photosynthetic enzyme that's found in red algae, is better able to distinguish CO2 from oxygen and therefore doesn't have this problem of photorespiration that we see with most land plants. But it has an even slower reaction rate than the reaction rate of the terrestrial plant rubisco. And apparently, this was analyzed by Long et al., and apparently this trade-off is pretty widespread. So it will be very difficult to eliminate photorespiration without paying a cost of having to have higher amounts of this enzyme because slower reaction rate. And the enzyme is already, one of the main nitrogen demands of the crop. It's a very, very abundant enzyme in leaves already. So if you accept this, then whenever I see a much ballyhooed announcement that somebody's got, genetically engineered crop that's drought tolerant, I always expect there are going to be some trade-offs there, because drought tolerance is something that would have increased individual fitness over a wide range of past environments. And typically the change they've made is something like increasing the expression of an existing gene. It's not something more sophisticated than that. So I always sort of look in the papers for, how was the yield under well-watered conditions? And that's odd. They don't present those data. But this third category of trade-offs is an area where I think there's a lot of potential to make improvements. And the three examples that I'm going to discuss, I'm a little bit nervous about this, of course, given the expertise in the audience, but the trade-off between human fertility and longevity, where I think there may be potentials to both increase health in old age and also slow population growth in ways that well, I don't know, I'll get to that. A trade-off between the individual competitiveness of crop plants and the collective performance of the plant community, which gives things like yield and perhaps other important collective traits. So there we've got some potential exploiting those trade-offs to increase crop productivity. And then the area that I've worked on the most, the trade-offs between the fitness of symbionts like rhizobia and their plant hosts. And there's potential to improve nitrogen fixation and the performance of a crop, but also the possibility, I think, of improvements that last more than 1 year, improvements in the soil microbial community that would benefit subsequent crops. So those are the three cases that I'll be talking about today. So the first example, you're probably wondering what organic tomatoes have to do with fertility and longevity. But when I started as director of the long-term research at Davis, the first thing I did actually was to visit the long-term experiments at Rothamsted, England, which are shown here. They were celebrating their 150th anniversary, and I was excited to be part of that. And one thing that really impressed me there was their archival samples, all these old glass, lead-sealed containers of samples from, the late 1800s and so on. And so we made a real effort at LTRAS to collect archival samples for future research. And Robert Norris, a weed scientist, set up these weed preserves and collected weed seeds from year 0 or year 1 for use by future generations of evolutionary biologists to let them compare the evolutionary process in the 10 different cropping systems and go back and have the ancestral genotype preserved in these seeds. But we also collected, among other things, a few of our tomatoes. And just a couple years ago, Allison Mitchell looked at these archival samples, convinced herself that there weren't biases introduced by changes in storage, and compared flavonoid concentrations in organic and conventional tomatoes. And as you can see, these are roughly double in the organic tomatoes relative to the conventional tomatoes, and these are side-by-side replicated plots managed by the same people. And these flavonoids are thought to have various beneficial health effects. But the thing that struck me is these are apparently, at least one of their roles, is that they're plant defensive toxins that are induced by various kinds of pest or pathogen attack. And so that really made me wonder, why is it that these toxins are beneficial to human health? And it turns out that it's not an isolated example. That was sort of my first thought. It's just sort of, chance. But there's a wide range of plant defensive toxins for which there's at least some evidence that they're beneficial to human health. In low doses, of course. This phenomenon is known as hormesis, and there are also some other curious examples where things like sublethal high temperature stress can increase lifespan dramatically in some species. In some cases, things like oxygen stress can increase longevity. Now, Matson and Chang noted this widespread phenomenon, and their explanation was that these toxins induce stress responses which are beneficial and, in various ways, which is fine as far as it goes, but if these responses are so beneficial, you have to ask why they're inducible rather than constitutive. Why aren't they turned on all the time? If you guessed trade-offs, you win an organic tomato. So this is one of the chemicals. Resveratrol is one that's received quite a bit of attention. And as you can see here, there's an increase in longevity and an increase in reproduction late in life, way out there at 8 days of age. But early in life, there are decreases in reproduction, as shown here, here, and here. Now, reproduction is a risky process. There's direct risk of death associated with reproduction. But our hypothesis, which I'm not sure if it's right or not, seems plausible that in addition to the risks associated with actual reproduction, that there may be risks associated with fertility that are somewhat independent of actual reproduction. That the blood pressure that's optimal for reproduction isn't necessarily optimal for longevity, that hormone levels that are optimal for reproduction aren't necessarily optimal for longevity. And we know that's true for testosterone, for example. So that suggests that there should be trade-offs actually between fertility and longevity. If that's the case, what determines whether we have the levels that are optimum for reproduction or the levels that are optimum for longevity? Well, we've been looking, since this is the food section, we've been looking at food, and there's some rather interesting evidence that food, in addition to being a resource, is also an environmental cue. And so you've probably read about this, that dietary restriction can extend lifespan, as shown here. But it turns out that in both fruit flies and in nematodes, individuals that are on a restricted diet but smell food, that those food odors partially reverse this benefit. Now in humans, there's a rather curious result. I've seen this published in 2 independent studies, neither of them sort of, rigorous clinical trials. But both studies found that sugar-free diet sodas, which are sweet but don't contain any calories, are highly associated with metabolic syndrome. So maybe another example of food acting as a cue, not just a resource. So this all led to our shrinking gene pool hypothesis. And a key theoretical insight, which came from Hamilton, as key theoretical insights tend to do, is that when population is decreasing, we know that if population is increasing or stable, then fitness is increased by reproducing earlier. But if population is decreasing sufficiently rapidly, and if there's a sufficiently high risk associated with reproduction or with fertility, then delaying reproduction can increase fitness even if fecundity is lower. In other words, an individual might be contributing only 2 offspring over their lifespan rather than 3, but if they contribute those offspring into a gene pool of 10 individuals rather than 100 individuals, then their fitness is higher. So that's a key point. And so the hypothetical part is that food deprivation or consumption of plant toxins found mainly in famine foods predicted past population declines. Now, I don't know whether we're talking about past population declines in early humans or in primates. I'm not sure how far back we're going. Obviously, there would be sort of ongoing selection for correct responses to environmental information. And so our hypothesis is that we have inherited responses to these cues which trigger physiological changes that when a population is going to decrease, trade fertility for longevity. So these are the predictions of an individual-based stochastic model, 3 different genotypes, one that reproduces as early as possible. I think I've got it reproducing at age 3, something like that. One that doesn't reproduce until age 4, and then to test our hypothesis, a constitutive delay genotype, which when it receives some kind of cue that the population is decreasing, which we hypothesize could be things like eating leaves rather than eating meat and fruit. Under those conditions, delays reproduction, and not very much. Basically, what I assumed in this version of the model is that half of those individuals delay reproduction in years when the population is declining, and that doing so increases their chances of survival by 50%. So it's not a huge, dramatic trade-off. And you see what happens. Here you've got juveniles, with some juvenile mortality, and then these individuals start reproducing, follow the same path, subject to stochastic variation. But here, when the famine starts, these guys keep reproducing and pay a increased risk of death cost. This genotype also, these individuals also decrease in number, but because half of them are delaying reproduction, they don't decrease as quickly. And so they've become a larger fraction of the population by here when conditions improve, and take that over 2 cycles of famine, and they've basically doubled their representation in the population. So our thought, and this has actually been suggested without exactly the same theory. Yeah, so under this hypothesis, kale is good for you because it's bitter. So the potential benefits, longer and healthier lives, and also because population trends are driven by reproduction of young people and not by death of old people currently. In the short run, we'd actually predict a decrease in population growth from trading some fertility for longevity. Now, there are certainly some risk. I mean, even assuming this is actually, the hypothesis is correct and this is feasible and so on, there's some risks. We might have to give up teenage pregnancy, but also, you know, we would expect changes in age structure, and that would cause all sorts of consternation and you know, require various social changes and so on. But anyway, that's the first idea. Now, population growth is, I would argue, the number one challenge facing agriculture. It's certainly not the only challenge. But I've plotted here world grain production and world population, and I've scaled it so that they overlap when grain production per capita is at the recommended dietary supply. Now, this obviously leaves out, you know, fish and you know, grazing and so on. But just looking at grain, there have only been a few years when we actually managed to exceed that recommended grain production per capita. Now, population is expected to increase, perhaps on this trajectory, perhaps on this trajectory. And the question is, what's going to happen with crop production? Now, if we don't deal with the problems of ongoing evolution in weeds and pests, we could actually see crop production going down like that. Or if we don't deal with erosion and all the various other sort of sustainability issues. But just solving pest problems isn't necessarily going to give us the substantial increases in productivity that we need if we're going to maintain food security without cutting down all of the tropical forests and draining all of the wetlands for food production. Now, yields have been increasing still in many parts of the world, as we see here for maize yield in Nebraska. But much of that increase is due not to development of improved varieties and not to development of improved practices, but rather wider adoption and diffusion of the best practices that are already out there. And you can see that if you look at the yields of contest winners for irrigated maize, where the highest yields recorded in Nebraska were 20 years ago. The cultivars developed now are not outperforming the ones that were developed 20 years ago under, you know, really intensive expert farmer management. Now, if you look at side-by-side comparisons, it looks like there's been a lot of genetic progress, improvement in crops. This is the yield. This is IR8, the original Green Revolution rice variety, and in the side-by-side comparison, it yielded less than IR72, a more recently developed rice variety. And that's interesting. This was true over a whole range of nitrogen fertilizer rates. In a side-by-side comparison, this is IR72, and this is the IR8. But back in 1968, when IR8 was released, they did the same experiment. And back then, not only did IR8 have higher yield than IR8 has today, it also had higher yield than IR72 does today. It's just that the pests that attack IR8 have had 30 more years to evolve the ability to attack IR8 than they have to attack IR72. So we need to continue what I call red queen breeding, keeping ahead of evolving pests. But we also need to move beyond that and look at yield potential, that is, improvements in yield under pest-free conditions. And the most promising approach, until we get to the point where we can actually, design Rubisco from ground zero, we might have some great stuff there. What I see as the most promising approach is what was done during the Green Revolution, and it's an approach that was laid out very nicely by the Australian agronomist Colin Donald 42 years ago, and based on trade-offs. And what he said is there's often a trade-off between the competitiveness of individual plants and the collective performance of a group of plants growing together, because after all, a plant that's competitive is likely to suppress its neighbors, and its neighbors in a system with good weed control are fellow crop plants. So he laid out that theory and some specific traits, of which I've listed some here. The first is short stature, and that was the primary basis of the increases in yield potential of Green Revolution rice and wheat. He also noted that most wheat varieties have multiple stems, as shown here, and suggested going to a single stem. Now you can see that, maybe you can't see, but these plants may actually produce less grain per plant. Donald's plants may produce less grain per plant, but you can fit more of them per hectare. And so you get more grain production per hectare. I've actually looked at the relationship between stem number and yield, and it's in the direction that Donald said it would be. I'm not sure about going all the way to single stem. There's some issues there with planter skips and so on. But at least his sort of qualitative analysis seemed to be correct. And then the third one, which is more relevant to the experiments I'm going to tell you about, is that the leaves should be relatively erect rather than horizontal. Now, again, there's an issue here of individual plant versus community. A plant with erect leaves, you can see there's a photon here that's not being intercepted by this erect leaf cultivar that would be captured for photosynthesis by a horizontal leaf cultivar with the same total leaf area. But you can fit these erect leaf plants closer together without mutual shading. So again, natural selection is primarily about individual fitness, but we're really concerned in agriculture about productivity per hectare. Now, photosynthesis, as you know, saturates at high light, and as a result, these erect leaf plants tend to spread the available light over a larger leaf area. So if we do that, if we take this amount of light and spread it over twice as much leaf area, we get slightly lower photosynthesis per unit leaf area, but we have double the illuminated leaf area, so higher total photosynthesis. So some of Donald's ideas have been implemented, some of them have not, but I'm going to tell you about sort of an extension of his ideas that's work that we did in my lab, and that has to do with solar tracking. So this leaflet here has slightly higher photosynthesis because it's aimed right towards the sun. But this leaflet here is being shaded by a leaf above it, which is also tracking the sun, and because it's tracking the sun, it's casting a larger shadow. And so computer modeling that we did suggested that this cost actually outweighs this benefit if you look at it on a plant community basis. So to test that hypothesis, we built this outdoor photosynthesis chamber where we could measure photosynthesis of a community of lucerne plants on a turntable, and we had a little crank outside. So we could basically measure the photosynthesis and the light interception with plants doing their normal solar tracking thing, and then turn the plants to disrupt solar tracking and see how that affected photosynthesis. Now, I had a paper rejected because I used crop rotation in the title, and they didn't like that. But this is actually this work's going to be in the Evolutionary Applications article we'll be submitting shortly. This is the computer model. These are some of the experimental data. And this is set up as the ratio of photosynthesis and the tracking to disrupted mode. So a value above 1 is a benefit to solar tracking, and below 1 is a cost of solar tracking. And you can see that at low leaf area, there is a predicted and actual benefit to solar tracking, but not much, 2%. I was surprised it was that low. And at higher leaf area, there's a photosynthetic cost to solar tracking, again, relatively small, which suggests to us that the selection for this trait may actually have more to do with shading neighbors than with a photosynthetic benefit to the tracking plant itself. Now you can see there's quite a discrepancy between the computer model and the data. I mean, here we have a break even at leaf area 2, and here the break even is up at leaf area 4. So quite a discrepancy there. And when you have a discrepancy, obviously the model is wrong. The problem is, which model? Because of course we have a model community here, and this model community has much greater edge effects than you would see in the middle of an alfalfa field. These lower leaves are getting light from the side that they wouldn't get if they were in the middle of a field, and that means the cost of shading by upper leaves may be less in this experimental model than it would be in the field. Now, that's obviously something that would need additional experiments to confirm or reject that hypothesis. But I'll just tell you that when I ran this model over the whole growing season for Davis, we actually got 5% higher seasonal yield if we got rid of solar tracking. So there's a possible idea. We could certainly breed non-tracking alfalfa, and there are other crops that have this behavior, some cotton cultivars, for example. There's a potential benefit of increased yield potential, but we would expect these crops to be less competitive against weeds. Now that's also true, of course, of, the short Green Revolution varieties. It's true of all of Colin Donald's suggestion. And so the question is, how big of a yield benefit do you need to accept the need for more effective weed control? And I don't really have an opinion on that. But let me move on to my third and final topic, trade-off, and that's the trade-off between symbiont and host fitness. I know some of you are familiar at least with some of this work already. But I wanted to start by making the point that food security is, job number one for agriculture, but we're also very concerned about environmental impact. And so we know that fertilizer use in the American Midwest is making a major contribution to the anoxic zone in the Gulf of Mexico. And it's, a multidimensional problem, but it's possible that increased use of biological nitrogen fixation could contribute to reducing that problem as well as various other problems. But there are some constraints on that, constraints in some cases that are better recognized in Australia than the US. And one of those is that rhizobia vary greatly in how beneficial. Different rhizobium strains that infect the same host. In this case, they were taken by Jeremy Burden's group from the same soil. And we see almost tenfold differences in plant net benefit from different strains from the same soil. Obviously, what we'd like to have in our plants are the better strains. Now, this is net benefit, but we've been thinking recently about separating out costs and benefits and going into that into a little more detail. And so this is one way of doing this, plotting as a proxy for cost, nodule weight per plant, and benefits. This is seed production, so it's not only agricultural yield, but arguably a direct measure of fitness as well. And each of these data points is a different genotype. These are old data from Abel and Erdmann, who didn't plot them anything like this. But you can see that these three strains here provided essentially no benefit to the plant, but they also occupied very few nodules and had a very low cost to the plant. And we can distinguish that from this. So I would call those losers rather than cheaters. But over here we have what I would call a cheater, a strain which is providing relatively low benefit to the plant relative to these main sequence stars, rhizobia, up here. Here's a strain that has the same nodule cost, but double the benefit. And over here is our super altruist strain, which is the popular inoculum strain USDA 110, which for some reason tends to not persist in soils. Now, we're also interested in, as I said, measures of efficiency, the ratio of benefit to cost. And there are at least a couple of ways of looking at that. One cost, of course, is the construction cost of building a nodule. And so we're looking at nodule weight as a cost. And these are data from my student, Ryoko Ono. And it turns out that there are a few rhizobia that have a wide host range. And so in this case, it's the same rhizobium strain providing more benefit relative to cost in P relative to in bean, based on the construction cost. But there's also the cost of operating a nodule, which we measure as respiration. And so here we have the slope of this line is the nitrogen fixation per unit of respiration. And this is a method that was worked out by the late John Whitty, who I was lucky enough to work with briefly many years ago, and we're now resurrecting it. We don't have bean on this. This is data for P. We don't have bean on this graph yet, but we're very interested to see if there are differences there. So if there are differences in the efficiency of nitrogen fixation, one of the reasons, some rhizobia are just defective. But there are also ways that rhizobia can divert resources from nitrogen fixation, which benefits the plant and incidentally benefits any competing rhizobia on the same plant. Divert resources from nitrogen fixation to their own fitness. And an obvious way is just to reproduce, but here's another. Polyhydroxybutyrate, or PHB, is this energy reserve found in rhizobia. And here we have wild-type nitrogen-fixing rhizobia and a little bitty granule of PHB. The only difference in these rhizobia on the right is a one-base knockout in nitrogenase, which stops them from fixing nitrogen. And that simple change diverted large amounts of plant carbon into polyhydroxybutyrate, as you can see here. So that creates a potential conflict of interest between rhizobia and plant, assuming, and I've assumed this for years, assuming that the PHB is actually beneficial to the rhizobium fitness. My grad student, Will Ratcliffe, who was also the brains behind the shrinking gene pool hypothesis, set out as his first experiment to test this hypothesis. And what he did was to generate 3 different populations of rhizobia that were genetically identical but differed in the amount of PHB per cell, and found that in a medium without any available carbon, that these rhizobia can use this PHB as fuel to at least double their numbers and also to survive, actually for over a year, it doesn't go that far in this graph, to survive for over a year without any source of carbon, whereas the same genotype starting out with lower levels of PHB didn't reproduce and died off more quickly. So there is a fitness benefit to the rhizobia from having PHB, and therefore a conflict of interest between the host and the symbiont as to how much PHB they should make. Well, I'll have to tell you about this. This is work that Stuart West did on a brief sabbatical. Some of you may know him and may not be surprised that he put out 2 papers in 2 or 3 months in my lab. But so the question that he asked is, given that fixing nitrogen does feed back and provide some benefit to the rhizobia, because the healthier the host plant, the more photosynthate it can provide, but that comes at the expense of not making PHB or reproducing more in the nodule or whatever. Given that, how much nitrogen should rhizobia fix? How much should they invest in nitrogen fixation? He tried to model the evolutionarily stable strategy for rhizobia, and basically looks like this, where this is the investment in nitrogen fixation, and this is Hamilton's R, sometimes called relatedness. So if you had a plant with only a single strain of rhizobia, and they invest in nitrogen fixation, the leaves have plenty of nitrogen, they can photosynthesize more, they benefit from that, and that predicts, under those conditions, a substantial amount of nitrogen fixation being an optimal investment. Now, if there's plenty of available nitrogen in the soil, then as you might expect, the optimum investment by rhizobia in nitrogen fixation is less. But that really doesn't matter, because real plants look like this with 6 or 10 different strains per plant. And under those conditions, the model predicts investment in nitrogen fixation of 0 as being the evolutionarily stable strategy. Why fix nitrogen that's going to benefit other rhizobium strains, which are not only potential competitors, but your most likely competitors for the next crop, because you're right, there, and they don't move very far. So this model predicted that nitrogen fixation should have died out 49 million years ago, which hasn't happened. And so the second, the hypothesis that I wanted him to test was to ask, what happens now if the plant monitors individual nodules, and based on how much nitrogen they're fixing or how much they're costing, does something which is adaptive from the plant's point of view, but for which from the rhizobium point of view could be seen as a sanction, something that reduces the fitness of rhizobia that fix less nitrogen. And so he modeled that, and that's just as bad, but basically this is essentially a horizontal line showing that if you put host sanctions into the model, then regardless of soil nitrogen level and regardless of the within host genetic diversity, you get a substantial investment in nitrogen fixation as being optimal. But that, of course, begs the question, can plants do this? Now, do they actually have the ability to monitor individual nodules, and do they then respond in some way that affects rhizobium fitness? So to test that hypothesis, there's actually was earlier evidence that they probably do based on comparisons among strains, but of course with comparisons among strains it's hard to be sure that the plants aren't responding to some arbitrary signal as opposed to actual performance. I used the analogy earlier that when you're thinking about plane hijackers, you should ask, do we want to keep people with certain names off airplanes, an identity-based approach, or do we want to keep people with knives off airplanes? So we know that plants respond to arbitrary signal molecules and exclude certain strains based on that, but do they also have a performance-based sanction? And so to test that, we used a nitrogen-free argon-oxygen atmosphere with just enough nitrogen gas in it to meet the nitrogen needs of the rhizobia themselves. And we did that either with a split root system where half of the nodules were prevented from fixing nitrogen, or with these individual nodule chambers where individual nodules were either in a argon-oxygen atmosphere or in a normal air with nitrogen. And consistent with the sanctions hypothesis, the rhizobia prevented from fixing nitrogen reproduced only about a third as much as the same genotype that was fixing nitrogen. This was largely due to differences in nodule growth, although not entirely. Now, ignore that X which moved. So this is sort of at the stage that our work has got into, textbooks and so on. But there's an interesting evolutionary problem here that we're working on now, and that is differences in life history of the rhizobia. Now, all of that work that I've showed you so far has been with soybean, and in soybean, when the rhizobia start fixing nitrogen inside a nodule, they don't change very much in size or shape. And when the plant dies and the nodule senesces, they are still reproductively viable and presumably escape out into the soil and form the next generation of rhizobia. But in a number of other legume species, including pea and lucerne among the crop plants, and clovers would be another example, there is rather extreme plant-imposed swelling of the rhizobia. As they acquire the ability to fix nitrogen, they swell up, sometimes they become branched, and they become very susceptible to osmotic shock. And the evidence is fairly convincing that when the nodule senesces, all of the cells that were fixing nitrogen, or at least the vast majority of them, die. And the next generation of rhizobia in the soil are not descended from cells that ever fixed nitrogen, but rather from their clone mates who had not yet differentiated into the nitrogen-fixing form. And so we, of course, saw some analogies between this and the social insects with non-reproductive workers and reproductive queens and so on. And we had a paper in New Phytologist last year discussing some of the possible evolutionary implications of this, because, for example, if you're not going to have any descendants, there may not be much point in hoarding PHB. And in fact, what you see in many of these cases is that these rhizobia hoard PHB and these rhizobia hoard PHB, these reproductives, but these don't. So if that's the case, if that avenue for cheating is foreclosed, by this host suppression of reproduction in the nitrogen fixing differentiated form, do these hosts still need sanctions? We went through some arguments suggesting that they might very well, in the sense that there are probably a lot of rhizobia out there that aren't necessarily cheaters but are nonetheless defective, and plants would benefit from not wasting resources on nodules that contain those less beneficial strains, even if the less beneficial strains weren't benefiting from being less beneficial. But it's not totally obvious that the plant actions that would prevent plant resources from being wasted would necessarily reduce the fitness of the rhizobia. So we've been repeating these experiments, and so far in P, there's clear evidence from repeated experiments of sanctions similar to what we saw in alfalfa. But curiously, or lucerne, but curiously, in P we see sanctions similar to what we saw in soybean. But in our experiments so far with lucerne, we haven't seen any evidence of host sanctions. So we're doing some more work on that, but that'll be very interesting if it turns out that lucerne doesn't impose sanctions. Now, this trait, which I've called sanctions from the rhizobium point of view, presumably evolved or was maintained by selection, or was selected for because of its immediate short-term benefits to individual plants. That plants that shut down nodules that aren't fixing nitrogen free up resources to do something else. But there would, of course, be effects on the soil rhizobium community that would affect subsequent generations of legumes. And so you can imagine this patch of wild legumes and a plant's neighbor saying, you've got some mediocre rhizobia there that are, fixing a little bit of nitrogen but not much, kill them so that our progeny won't have to deal with those bad rhizobia. And the plant in question responding, I need this nitrogen. The net cost to benefit ratio to me as an individual plant of supporting these mediocre rhizobia is positive. What has future generations ever done for me? But that's sort of the same kind of argument that Colin Donald would have made for shorter plants or so on. So we as humans, those of us that are plant breeders, we can certainly select for things that natural selection wouldn't, and one of those would be benefits to subsequent crops. And so here's just sort of a rough sketch of an approach of how that could be done. You start out with these are all drawn the same, but a genetically diverse population, a population that we expect may have some sanctions variance in it, and they're growing in soil with a mixture of a bunch of different rhizobium strains differing in mutualism. And so we grow them and we save the seeds from that first crop, and then we select from among those seeds based on the performance of subsequent crops grown in those same pots. And what I've shown here, because Nancy Johnson has published data suggesting that soybean can have significant effects on mycorrhizae that benefit maize, what I've shown here is two genotypes in the original population that have benefited maize, which typically will be the next crop in the rotation in the Midwest. And so seeds from those plants are retained. The other ones are discarded in this highly simplified scheme. And then of those two plants, one of them, because it imposed such draconian sanctions and only let the very best rhizobia in its nodules survive to recolonize the soil, that plant there also had this benefit to the another soybean plant 2 years later. And based on that, we would retain the seeds from this for future assessment. Now, I've emphasized here benefits to future crops based on the soil microbial, the plant effects on the soil microbial community, but there are other possible benefits to subsequent crops which natural selection wouldn't give us, but which we could perhaps produce through an appropriate selection program. An example of that, I'm afraid I don't remember the author's name, but a seminar I heard many years ago by an Australian agronomist who discovered that a significant part of the benefit from lupins to a subsequent wheat crop wasn't the nitrogen fixation, but rather that the taproot of the lupin was bursting through a hard pan, creating channels which could be used by the subsequent wheat. So that's another example of the sort of thing that we could perhaps develop selection schemes to produce things that would be beneficial in agriculture, but which natural selection wouldn't have given us. So that's basically the idea, to breed for benefits to subsequent crops, missed by past natural selection, benefits such as more mutualistic soil microbes, but my assumption is that there probably would be at least some short-term yield cost in the soybean, for example, shutting down nodules with mediocre rhizobia that are providing some nitrogen but not as much as the best strains. But the long-term benefits might outweigh that. So just to summarize, I've talked about trade-offs between human fertility and longevity, presented a perhaps controversial hypothesis that we might exploit those trade-offs in ways that would increase our health and our old age if we're willing to sacrifice teen pregnancy. I've talked about food security in the context of increasing crop yield, and my thought at least is that over the next 10 or 20 years at least, the best opportunities for increasing yield potential are going to be when we really pay attention to trade-offs rather than assuming that we can get trade-off-free improvements. Someday molecular biologists may be able to deliver trade-off-free improvements to traits like photosynthesis and drought tolerance, but I don't expect it anytime soon. And finally, improved use of symbionts. I've been talking about rhizobia, but we know that mycorrhizae can be beneficial. We know that there are bacteria that live on the roots of plants and suppress pathogenic fungi. So those are all areas where the bacteria that suppress the fungi are not doing it for the benefit of the plant. So although we're getting some benefit from those bacteria, because there hasn't been selection for that trait, it's something that we could perhaps select for if it's something that can be manipulated by the plant, because we have control of the plants that are growing in our fields, at least in arable crops. So those are my thoughts, and thanks to the people who did the work. Thanks to NSF for keeping us in money all these years, and thanks to all of you for your attention late in the afternoon. Thanks.